pancreatic juice is what we call the cocktail of enzymes and salivary juices and bicarbonate that is released from the pancreas, right? It's not putting the pancreas into a blender and making juice, right? And then bile, we talked about bile. Bile is that liquid that flows from the liver to the gallbladder containing the bile salts, which are responsible for emulsifying fat. Now, the acinar cells of both the pancreas and the liver are going to secrete these substances. Acinar cells in the liver secrete bile, acinar cells of the pancreas secrete pancreatic juice, and And so the acinar cells have a smaller component in terms of the volume of this fluid, in terms of the volume of this secretion. This is going to be water, electrolytes, and some enzymes. And again, this is the smaller component of these secretions, of these juices. The duct cells, right, the cells of both the pancreatic duct and the hepatic duct, right, the common bile duct, the cells of those ducts also secrete components of these secretions. But this is going to be the majority of the volume, the liquid component of these secretions. And it's also going to be bicarbonate rich. Why is it bicarbonate rich? Why do the duct cells of the liver and the pancreas secrete this bicarbonate-rich secretion? I sort of mentioned this yesterday. Right, this is to neutralize the acidity of the stomach. So when this substance that's been mulched and mixed in the stomach with that acid, when it moves into the duodenum, we need to neutralize it because the duodenum does not have that protective coat against the acid the way the stomach does. So we need to start to neutralize the contents. And so the large portion of the volume of these secretions is made from the duct cells containing water, containing bicarbonate, and then the smaller portion, which is secreted from the acinar cells, has those enzymes and electrolytes, etc. Okay. All right, let's move on to talk about CCK and secretin, two hormones that we mentioned as regulatory factors in the stomach. So CCK standing for cholecystokinin and secretin both have a very similar response. Their response is to secrete bile and bicarbonate into the duodenum. Now, the pancreatic juices have different contents. So the pancreatic juice at any given time looks very different because it's responsive to our diet. So if you're eating very acidic meals, your pancreatic juices or your pancreatic secretions are going to have more of a bicarbonate in that secretion. If you're eating more fatty meals, you're going to have more lipases in that secretion. If you're eating more protein, like a protein-rich diet, you're going to have more proteases in that secretion. So the pancreatic secretions are going to be tailored, customized, depending on exactly what's coming in your diet. Now, when we talk about CCK, CCK stimulates the acinar cells specifically to secrete their enzymes. These are the cells that are within the glandular tissue of both the pancreas and the liver. So CCK is telling these cells to release enzymes. Okay? It's also going to be a small stimulant for bicarbonate release, right? That's not its major function, but it's also going to help with bicarbonate release. CCK also potentiates the effect of secretin. We'll talk about what secretin does, but CCK basically gives secretin a push to do its job. I don't know how to explain that, but CCK is not doing the work, it's pushing secretin to do the work. So it potentiates the effect of bicarbonate release, which secretin is really responsible for. Moving on to secretin. So secretin is stimulating the duct cells. So you want to bear these two distinctions in mind. CCK acts on the acinar cells. The acinar cells are the cells, the actual glandular cells of the pancreas and the liver. And it's telling those cells to secrete enzymes. Secretin is working on the duct cells. So the pancreatic duct cells, the liver, the hepatic duct cells, and it's stimulating the release of enzymes, and more importantly, the release of bicarbonate. Okay? And again, you want to mention here that secretin potentiates the CCK effect on the enzyme release. So basically, we have two enzymes, CCK, secretin. They're doing their own job independently, but the other's also pushing the other to do its job. So secretin is potentiating CCK, saying, look, you got to do this. You got to release those enzymes. whereas CCK is potentiating secretin, saying you got to release bicarbonate. So along with their own individual jobs, they're potentiating each other to release their secretions. Let's talk about the stimuli for each of these. So the stimulus for CCK is going to be fat amino acids in the duodenum. Why specifically fat? Why specifically fat? All right, we'll get to that. So the stimulus for CCK is fat and amino acids in the duodenum, and the stimulus for secretin is the acidity in the duodenum. So excess acid is going to potentiate secretin. That makes sense because secretin's role is bicarbonate. So if you have lots of acid, we stimulate secretin. Secretin is going to release bicarbonate to neutralize that acid. All right, same thing for CCK. The stimulus for CCK is the presence of these macromolecules. So if we have macromolecules, we stimulate CCK. CCK releases enzymes to dissolve those molecules. Okay, so if you look at the stimulus, you can tell exactly the job that this hormone is doing just because of its stimulus. All right, so let's look here at so let's look at a scenario, a flow chart here that describes those functions. Oops. So we have presence of acid in the duodenum. That comes with that acidic kind that's coming from the stomach. The chemoreceptors will detect that acid, and they will elicit that short and long reflex. Now, these short and long reflex will then elicit the endocrine system to stimulate the secretion of secretin. Secretin is then going to encourage the release of bicarbonate. What else did I say secretin is going to do? So it's going to, on its own, that's its own function, to release bicarbonate. But what else is it going to do?
Push CCK to do its job.
Good. It's going to push CCK to release enzymes as well. So it's going to potentiate CCK's response. And that's going to bring about the bicarbonate release into the duodenum, which will neutralize the acid in the duodenum. And this feedback does turn off. So once the acid in the duodenum is neutralized, we turn off that initial response, which again is the acidity of the chyme that's coming into the duodenum. Regulating bile secretion into the duodenum happens via secretin and CCK. So secretin is going to stimulate bile secretion from the liver. All right, we made that clear. Secretin is going to tell the liver to secrete more bile. CCK is going to tell the gallbladder to secrete more bile. Now we know bile is made in the liver, stored in the gallbladder. So secretin basically says you need to make more bile. CCK says you need to release more bile from the gallbladder. And it's going to stimulate gallbladder contraction as well as the relaxation of the sphincter of Oddi. Where's the sphincter of Oddi? We talked about it yesterday. Where exactly is the sphincter of Oddi? So the sphincter of Oddi we mentioned is regulating the release of the pancreatic duct and the common bile duct. So these two ducts are going to meet at the sphincter of Oddi, and depending on the relaxation or contraction of that sphincter, will we have the release of pancreatic juices and bile into the duodenum. All right, let's look at another flow chart here. So we said the presence of acid in the duodenum is going to encourage secretin release, and the secretin release is going to encourage bile secretion from the liver, specifically telling the liver cells to secrete more bile. Whereas protein digestion, the presence of those molecules, proteins, fats, amino acids in the duodenum, that's going to encourage CCK. CCK will then do two things. Relax the sphincter to open up that duct so that fluids can move into the duodenum, as well as encourage the gallbladder to contract. And the contraction of the gallbladder is what squeezes or secretes that bile out into the duct system. Okay. Now, together, all these effects basically encourage the emulsification of fats in the presence of bile and the neutralization of acid with the presence of bicarbonate. So this is what I meant when I said that secretin and CCK have a very symbiotic relationship. They're working towards the same goal. They use different mechanisms, but their end goal is the same: to neutralize the acidity of the chyme that's coming from the stomach and to emulsify the fats that are coming in the stomach as well.